can't wait 17 years for a solution. You need a solution that you can get out there safely in just a few years. And I think now biology is presenting us with some natural answers that are already there, an opportunity to do that.
Welcome to Labs Report. I'm Justine Levin-Allerhand. You may notice that our set is a little bit greener today. We're actually focusing on agriculture, so we wanted to give you a little bit of a surround that matches what we're going to be discussing. I'm here with Ryan Rapp. Welcome, Ryan.
Nice to be here.
You're the CEO of Terrana Biosciences and origination partner at Flagship. You lead a team of people who are originating original bioplatform companies in agriculture and sustainable resources. You've also held R&D positions, most recently as the CTO at Pairwise, agricultural genomics at Illumina, as well as other leadership roles at Monsanto. But before that, you grew up on a farm in Massachusetts. So eager to hear a little bit about your background and what brings you here today.
Yeah. When I was growing up, I was fortunate enough to live down the street from a family farm. And when I was about, I think, 14, which is the legal age where you can start working on a farm, I started working the summers down there. And the woman who ran the farm, Shirley Cahill, was good enough to explain everything to me, teach me about the pests, teach me how the practices were done to get a good harvest, how to start the plants. And that really sparked my interest in it. At the beginning. From there, it kind of just dovetailed into me wanting to pursue that some more. Went to Cornell for my undergrad degree in plant science, did my PhD at Iowa State, and then went on, got interested in agricultural biotechnology. Worked at Monsanto for about a decade, developing molecular breeding products and also biotech products, and just have absolutely loved every minute of it.
Turns out that agriculture and this conversation is a bit of a homecoming for me as well. My original research that got me excited about going into science at all was actually in college at Barnard College, working with Professor Brian Morton, where I was studying plant genetics. So it's exciting to be here today to talk about what's happening at Terrana Biosciences. Can you share a little bit about what Terrana Biosciences does? What was a bit of the origination story that brought us here today?
Yeah. So, Flagship has a very rich history of RNA and doing work and research in RNA. And the question sort of came up, can we apply everything that we've learned to this and what we know about the natural RNAs that are inside plants? And can we find a way to produce products that protect our plants from insects, bacteria, fungi, but do it in a way that's perhaps safer and more sustainable than using small molecule chemistries, which is the bulk of what we use today. So that was sort of the origination idea for it. And then the team was led into the discovery that in plants, and it's not fairly well documented in many other species, there are numerous types of RNA molecules that are benign that live inside the plants and are also in some cases beneficial, and they're not part of the genome of the plant, right? They're coming from external sources. And so it led to the idea that, hey, we can take these things that are already helpful to the plant and we can use them to deliver some of these solutions and start to create platforms where we can create many products to solve a fungal problem, an insect problem, a bacterial problem.
So take something that's already in the milieu and harness it in order to create
Yes, stuff you're already eating every day and just reformat it to have a more beneficial effect.
This is great. Crop health, we don't talk a lot about crop health, but it affects all of us because it completely impacts the food supply, and it's affects more across the population than any single disease. And obviously we talk a lot about how we're helping in human disease, but if you think about the human condition, crop health is critical. So what's new about the state of the technology? What do we see that we could create an unlock right now in this moment in time, especially if these are aspects of what's in the ground or what's on the plants and been around for a long time? What's different now?
Yeah, I think one of the big things that's different now is the technologies we have to address biological questions. So you think about, we had an age of physics, we've had an age of chemistry, and now it's really the age of biology where we've got much more advanced techniques that help us get to the answers to different problems much quicker, help us understand how biological systems are working. And then I think the neat part of doing it in a company setting at someplace like Flagship is translating all that into stuff that can be useful in the real world.
And as we think about the stressors in the environment, is that also adding to the need for innovation? Is that or
So I mean, the planet is different than it was 50 years ago. And if you look across just at the raw data, the rate of catastrophic events, whether they be wind, heat, lack of rain, too much rain, all of those have increased in pace. And we have a cropping system that is working to keep up with it, but it really can't, right? Our pace of innovation for biotechnology is often quite slow. To make a new chemistry or to make a new biotech, it often takes anywhere from 10 to 17 years to get to the market, and it can cost upwards of
Mhm.
250 to $300 million. And so how do you do that when you have a new problem showing up all the time? You can't wait 17 years for a
Right.
solution. You need a solution that you can get out there
Mhm.
safely in just a
Right.
few years.
Mhm.
And I think now biology is presenting us with some natural answers that are already there, an opportunity to do that.
Yeah, we talk a lot about programmable medicines for humans, but it really is programmable medicines for crop. And it sounds like the state of our crop health is not as good as it needs to be, just like the state of our human health is not as good as it needs to be. So we need to find programmable solutions. Going back to that bridge between human health and planetary health or crop health, Flagship actually has a track record of drawing from our experiences in creating modalities in human health to advance agriculture or planetary health, and vice versa. And we've done it in a few examples over the years that I think have been really impactful. RNA, of course, has been a human health story to many of us. And but tell me more how Terrana uses RNA in order to deliver the instructions that will, activate the plant's natural abilities without actually changing the genes. So not actually genome editing, but actually using RNA for a positive effect.
Yeah. So when you think about even our own health, right? Inside of us, we have in our guts, we have good bacteria and bad bacteria, right? We've all
Mhm.
come to understand this. And just in that same sort of analogous situation, plants in them have they have bad viruses, right, that cause economic damage and disease. They also have beneficial and good
Mhm.
viruses that live in them that have been woefully understudied. And so we've partnered with some faculty members that have spent their careers studying this and use that as the jumping off point to say, well, can we take these things that are already beneficial forms of RNA or don't have any effect on the plant and really turn them into a tool that we can use? A lot of the acceleration in this was really, driven by the fact that there was this very rich ecosystem of RNA research and
Mhm.
understanding that was already going on at Flagship, which really allowed us to do it much faster than we would have otherwise.
Yeah, I think we've had other instances where we think so much about pathological states, but we also need to think about positive states and
Yeah,
how do
they're
you
out there.
leverage the commensal viruses, things that are quite positive, and same in plants as you could see in humans. So, as we've talked about before in this setting, what Flagship does is, go beyond the scientific adjacencies, take scientific leaps, and then try to actually unpack that scientific uncertainty. So, take us back to the early days of Tirana in the origination. What really sticks with you when you were first trying to chase this question of can we improve crop health, and what are the tools that we have to improve crop health? What were either aspects of inspiration or scientific unlocks in your mind that said, okay, now's the time and we could actually do this and this is how we should go after it?
The first group of RNAs that we started working with were viroids, and they're a very restricted class of RNA molecules, and not much was known about them.
Mhm.
And there was this one paper that showed, really beautifully how they replicated, how they moved, how they
Mhm.
coexisted with the plants in many cases. And it also said in the paper, you can't engineer these, you can't put cargoes in them. They had
Mm,
found one little spot where they could insert
mm,
something. They said they tried everywhere else and it didn't work. And we had a RNA structural biologist at Tirana, and she said, I think I can figure out how to put more
mm.
and different types of cargoes in there. And so we started a little experiment with them, and then that turned into our whole first platform that
Wow.
actually allowed us to deliver active RNA molecules to plants.
Yeah, so that's very much kind of an operating principle here at Flagship, where we'll challenge dogma and not and really just to say, well, what if it's possible? Let's allow it. Let's suspend belief. Suspend the current understanding and say, okay, so if it is possible, what do we need to do?
To
And
make it happen.
then all you have to do is ask the question and kind of free yourself of the constraints, and then maybe you could parse out and come up with a plan. And then if it works, amazing. If it doesn't, that's fine. We'll go back
Yep.
to the drawing
Go to the
board.
next one.
Right, exactly. Can you paint a picture of what the lab actually looks like? And, where does the work actually happen? We're so accustomed on the human health side of just what a series of lab bays look like. When you're doing, this type of work, what's the sequence of activity? Where does it happen? Is it just a series of folks at the bench and lab coats? What does that journey look like?
Yeah, in many ways, if you can step back a little bit from what we do and what's done in human health, there are a lot of similarities, right?
Mhm,
So we begin a lot of our assays in cell culture.
mm,
We
mhm,
typically use protoplasts and instead of a straight cell culture. That's where you dissolve the cell wall away from the plant. We get molecular readouts from those assays because they're very quick. We can learn how RNA is functioning, if we're expressing a protein well, all of those sorts of things. But then sort of the next step we can do a little bit faster, right? So after that, you might be going to a clinical trial in a human study. For us, we go into a greenhouse trial
Right.
or a growth chamber trial, right? So we'll migrate that
Mm.
technology first into a greenhouse or a growth chamber. That's stuff that we can do under permit within the facilities we have. And then when we're ready and we're seeing the type of efficacy we're looking for, we typically move into a field trial at that point. And so then we'll do permits with the USDA and the EPA to take that technology out to the real world.
Yeah, it is just so interesting because then we'll now talk a little bit about what you did. We think so much about cellular systems and animal systems and what do you need to do to model the disease state. Here, you went into working, you worked in eggplants.
Yeah.
So tell me about, what's the virtue of that model and what did you do here and how does that give you kind of the reasons to believe that you could then transfer it into other more challenging, areas.
Yeah, so this a story I was telling you before about that
Yeah.
paper was actually an eggplant.
Oh,
And so that was
mhm.
going to be the starting point for us from the perspective that there was a natural system.
I
We
see.
had already learned a little bit about the natural system. We knew how to grow eggplants in the growth chambers and greenhouse, so that was a good start. And we knew how to work with them from the molecular perspective because they're related to a lot of plants. There's some resources in. And so that was the place we decided to choose to say, can we make this work in terms of can we get an active RNA molecule inserted? Can we make sure it's circulating throughout the plant? Is it giving the type of coverage and protection that you would need throughout the life of the plant? But, from there, we have to take it and translate it into something that is going to be useful across a broad range of plants.
Yeah, so eggplant is the model,
Yep.
and then you had to actually pivot then to say, "Okay, if that's the model to determine the technology that's needed and to test the technical unlocks," you then have to actually go into a real crop problem, and you went into, tomatoes and controlling, brown rugose virus. And I was not familiar with this virus until, reading up on it in speaking with you and learning about it from the data. So tell me more about that threat and the history of that threat, where are we at right now, and, the results that you went after, through the Terrana technology.
Yeah. So when we were ready to start understanding where we thought the technology could have the best impact first, tomato came up and brown rugose came up. One, because it's partially related to eggplant. And so that was a
Mm.
good starting point.
Mhm.
But brown rugose disease is currently across the globe. And if you get it in your
Mm.
greenhouse, you can lose up to 75% of the plants that are in there. And you got to think about
Wow.
these greenhouses grow those tomato plants for 9 months at a time.
Mhm.
So that it's a huge loss for them.
Mm.
And then the 25% that are left, the fruit is typically so deformed that it's not marketable. You can't
Right.
sell it. Even if you go into the grocery store today and you kind of root around, you will find tomatoes that have
A
this
little bit.
a little bit of the mark on it. And in fact, when the CDC is benchmarking different types of human pathogens through molecular tests in sewage, one of the standard controls they use
Oh,
is brown
really?
rugose because
Yeah.
it's everywhere.
Yeah.
And so they
Yeah.
use that to benchmark against because you're eating it every day. The disease itself, there's some genetic control that's being developed by the seed companies, but what it's doing is it's forcing producers to not grow the varieties that they love in
Mm.
terms of flavor profiles that the consumers like, the colors, the timing that works in their operations, and other disease resistance pieces. And so they're really looking for a solution where they can grow the types of things that work well in their company production systems, but gives them that flexibility instead of being locked into a particular type of germplasm. So we decided to focus in that area first.
Mhm.
And we have a discovery pipeline that allowed us to quickly assess if there were the types of RNA elements in different tomato germplasm that we could use to do this sort of engineering. And fortunately, we found a whole bunch and we quickly settled on one and began product development.
So part of product development is actually ensuring that you could, really advance the technology, advance the product in a really seamless manufacturing process. Tell me more about what that looks like in your world and what that manufacturing process, the criticality of it, what were the challenges that you anticipated, things that you didn't anticipate. Once you figure out what needs to happen, you actually then have to deliver at scale. What does that look like
Yeah.
here?
So RNA is interesting because if you think about RNA for human applications, it is very expensive to produce and it requires a cold chain to keep with it, but it's enormously efficacious, right? Uh, We don't have a lot of cold chain solutions in agriculture, especially when you get to the growers out in the field or in their greenhouses. And we also have a very different price sensitivity, right?
Yeah,
Maybe you're treating a cornfield with 36,000 plants in it, but you can't spend more than like
exactly.
50 to
Mhm.
$100 to treat that whole acre.
Yeah.
And so we had to find a manufacturing process that was incredibly cheap for our RNA. We looked at some different ones. We were able to do some fermentation in E. coli, but what we actually settled on was producing these RNA particles in other plants. We actually found a
Mm.
way to produce them to an extraordinarily high amount in that
Mhm.
individual plant, and then we use the sap from that plant in the
Wow.
manufacturing process to actually
Yeah,
create
yeah,
the final product. The neat part about this is that
it's
these
really nice.
molecules are so potent that one plant producing this goes a very long way in terms of treating a couple of acres.
So we've been talking a lot about, crop protection, but what else does this technology do to help plants?
Yeah, we started with crop protection because from a company building and a business building perspective in agriculture, demonstrating that a crop protectant agent is working the way it's supposed to, that it's preventing disease,
Mhm.
is a lot more straightforward than showing that something can uh,
Right.
improve yield, right? Or
Mhm.
make those other things
Yeah,
better. So
exactly.
we started to build the business
I
around that.
see.
We also understood the willingness to pay there. But the reality is the technology base can deliver much, much more than crop protection. So as we build this business and we get working products in tomato, we're already looking at a host of different things we can do in tomato now that affect things like sweetness, help with plant architecture for harvestability in different greenhouse situations, control the color in
Mm.
ways, and this, I know it sounds kind of funny and trivial, but actually, when you get those packs of tomatoes, that's an enormous logistical feat because everything that's a different color, it ripens at a different time, and the growers have to stage
I
everything
see.
they're doing so that every day they can fill one of those things for you. They would love to have a variety that has multiple colors and matures at the same time, and we believe that's the sort of thing that we can help them with.
Yeah, it's really amazing to see that you went after just protection and then you create an enhancement
Mhm.
opportunity
Yeah.
too, which is really unusual. So, we talk a lot about we talked earlier about going into uncertain spaces, going into areas where you're really fighting against central dogma that, if people feel like it's truly impossible. Tell us more about that journey of other challenges you expected.
Yeah, I mean, I had to think of like a sort of like a high and a low point.
Mhm.
On the high point, one of the most amazing times was when the scientists were we were working in the lab and they were actually showing that these could circulate in the planet. It actually worked the first time the way that they expected it
Mhm.
to, which almost never happens,
Right.
right? So that was a great high point, gave us a lot of confidence to
Yeah.
go forward. And then I remember the first time we tried to express proteins off of these types of RNAs, and that did not go well at all. And that actually led to a revamp of how we thought about using the RNA molecules, a
Hmm.
different screening program. And it took that screening program and some insight from three-dimensional structure and engineering to get that all working. But in the end of the day, it worked. But it was a lot of iterations and a lot of
Yeah.
turns.
So for folks at Flagship who are pioneering or others who are going in white spaces, what advice do you give? And this doesn't necessarily be in agriculture, but again, you're an experienced originator and pioneer, and share where you think where people should be thinking from a mindset standpoint, from an activity standpoint, etc.
Yeah, I mean, I think the most striking thing I've seen at every place that I've worked is that the companies and the ideas that win are often those that are willing to stick with it. And what I mean by that is that you're going to get bad data, things are going to go wrong, and you can't just turn around and chase the next shiny thing that is right adjacent to it or somewhere nearby. You have to have backup plans for sure and be pursuing other things, but you also just can't give up and try to take one of those things the first time it goes wrong. You really have to be a little bit dogged about making it work and think about how you build your research organization so that they are resilient to that type of failure and that they have the type of pipelines that can help you work through that.
Yeah, I mean, so much about pioneering and leaping to a new location is actually having your eye on that future state
Yep.
constantly, such that when you have setbacks, you're still propelling yourself forward. And that persistence, I think, that
Yeah.
you're describing is ever present.
And make your experiments tell you things when you get negative data,
Yeah,
right?
that's right. That's right.
Set up experiments where you're going to learn from the
Right.
failures as well.
Yeah, absolutely. Ryan, thank you so much for joining. To learn more about Terrana Biosciences and to see the full data set, please go to our Substack, The Labs Report. Thank you for joining.